Welcome to the last screencast on the genetic engineering topic. This particular screencast concentrates on the Human Genome Project. Before looking at the Human Genome Project, we first of all have to consider how a piece of DNA can be sequenced. We are able to sequence bits of DNA up to about 750 base lengths long. Okay, and it was first of all discovered by a scientist called Sanger, Frederick Sanger, and sometimes therefore it was known as Sanger sequencing. It's also known as chain termination sequencing for reasons which will hopefully become clear in a second. So the process involves polymerase chain reaction, okay, with a slight modification. So if you remember, you'll note some polymerase chain reaction. A polymerase chain reaction is when we had a length of DNA, which we then added a primer to, which was a length of DNA which was specific or complementary to the DNA that we're looking at. We added the DNA polymerase and we added free nucleotides. We then heated up our mixture and cooled it down. Every time we heat it up and cool it down, the amount of DNA doubles. Okay, in a very basic summary, but if you're unclear on that, you need to go back to the screencast, which explain that in more detail. Okay, the slight difference here is that as well as the free nucleotides we add to our mixture, we add some modified nucleotides, and these modified nucleotides are modified such that whenever this nucleotide binds to its complementary base on our DNA, when we're replicating our piece of DNA, it stops the replication process or terminates that replication process. As well as doing that, it's modified with respect to the fact that we put some sort of fluorescent marker on our nucleotide so that we can detect when this termination has happened. Okay, so we put all these things into our PCR machine. Okay, and we have to set up 4 tubes in our PCR machine, 1 tube for each modified type of base. Okay, so modified adenine in 1, modified thymine in 1, modified guanine in 1, and modified cytosine in the 4th one. Okay, what we then do is that we put it in the PCR machine and we heat it up and cool it down many times so that many copies are made. Okay, but every now and again, one of the copies is going to be disrupted at some point. Okay, because one of these modified nucleotides is going to come in and bind by complementary base pairing to its complementary base, and then stop the replication, which means that we're going to get lots and lots of different sized bits of DNA produced. Okay, so that's quite complicated to understand that, so I shall try and illustrate that diagrammatically. So here we have a piece of DNA, okay, which we are wanting to sequence. Okay, and in this respect we are interested, in this particular example, we're going to sequence wherever guanines occur. Okay, so whenever guanines occur, they're going to bind to cytosines, okay, by complementary base pairing. Okay, so in this case, if we've added our DNA into this tube here, we've also added our primer. Okay, here's the primer here, our primer. We've also added our DNA polymerase. We've added free nucleotides, and then we've added these modified nucleotides, okay, which are represented here by DD cytosine. Okay, so what happens is that once all of these are in our tube, and our tube is in its PCR machine, we then heat up the tube, cool it down, heat it up, cool it down. Every time we do that, the DNA gets replicated. Okay, but every now and again, rather than a normal cytosine binding to a guanine, what happens is that one of these modified cytosines binds to a guanine, and when that happens, the whole process stops. Okay, so rather than the whole piece of DNA being copied, okay, we only copy up until the point where we've encountered that guanine. Okay. We then heat these things up and cool them down again, and at other points, other modified cytosines, okay, will complementary base pair to other guanines, okay, and here's again we have another one which is complementary base pair to there. So what we end up with is fragments of DNA which are all of different sizes, depending on where in that particular copy the modified cytosine has bound by complementary base pairing to its guanine. Okay, so we've got these three different fragments. Okay, what we then do, sorry, just before I say that, what we also do, obviously we're doing the same in 4 different tubes, okay, but this other tube is going to have the modified base thymine in it, adenine, guanine, etc. Okay, so we're going to end up with 4 lanes on our gel, one for the cytosine, one for thymine, one for adenine, and one for guanine. And using the guanine one, so this has been the one which has had the modified cytosine in, okay, so identifying where what all these are, this here is the shortest segment of DNA. So in the gel electrophoresis, it's going to go the furthest, okay, so it's going to be that one there. So we've identified that one there, okay. This one here is the next shortest, so that's that one there, whereas this one is the longest, biggest piece of DNA, so that corresponds to that there, okay. So, and also we've done the same for wherever the thymine is bound to its adenine, and the adenine bound to thymine, and guanine to cytosine. So we get this pattern, okay, and you can see all these bits of DNA, all different sizes, okay. So what we can do is that we can therefore work out that there is that piece of DNA there, the shortest one, okay, was the one with the adenine on it, okay, which is base paired to the thymine, okay, and we can work out the sequence, okay, if you like, here, 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 here. The sequence of these bases that occur here gives us the sequence of this piece of DNA, okay, or a complementary sequence rather to the piece of DNA that we're of interest, okay. So that's how the Sanger sequencing occurs, okay, and if I were to show you a little clip of that. So here's our primer being added. Primer being added to our single-stranded DNA. We've put those into our 4 tubes, okay, and then what we do is we add all our free bases in there, and then we add our DNA polymerase in here as well. DNA polymerase is being added. Okay, now we're adding our modified bases. Okay, so these are modified bases. So they've had these have just had extra phosphates added to them, which means that they don't form the sugar phosphate backbones, and therefore they terminate the whole sequencing process, or the whole replication process. So that's what they are, the modified ones. Okay, so we've got in these tubes, okay, I've got my DNA to be sequenced, I've got DNA polymerase, I've got lots of free nucleotides, okay, and I've also got modified nucleotides, one particular type of modified nucleotide in this tube, one in here, these 4 tubes representing the 4 different bases, A, G, C and T. So looking at one of these, the replication process is happening, and in comes a modified nucleotide, okay, which is bound by complementary base pairing to this base here, and the whole process has been terminated. Okay, what we then do is continue replicating that, and what we end up with is different segments of DNA which are all different lengths depending on where the modified base has terminated the whole replication process. Okay, then what we do is we add our mixture into our electrophoretic gel, which again we've studied before. And all the fragments are separated on our gel. Okay, so what this shows us is that all these different segments, okay, so here's the lane of the adenines, here's the lane of the cytosines, thymines, guanines. Remember that the shortest bits go furthest. Okay, so here's the shortest segment, the next shortest segment, next one, next one, next one, next one, next one, next one, next one. Okay, so that gives us the sequence here, C, G, A, G, T, C, A, G, okay, and obviously that is the sequence of bases that have done the terminating, okay, and they are complementary to these bases here, okay, so that will give us the actual sequence of the DNA that we're actually looking at here, okay, so that's how that's done, okay. For interest's sake, I'll let this run, and you can have a look at how another method of how that is done also. So this is called automated DNA sequencing, where all of the different fluorescent markers and modified bases are put into the same tube, okay, but there's a different fluorescent marker on every base. So here we've got our single strand unknown, we've got our primer's added, okay, the DNA polymerase is added, this is all in the same tube. We've got our bases are added, okay, so that's thymine, adenine is thymine, and then guanine, oh sorry, cytosine, and guanine are added. And then here we have the modified and fluorescent cytosine, the modified and fluorescent adenine, modified and fluorescent thymine, and modified and fluorescent guanine. All this is put in the PCR machine. Replication occurs. Okay, termination occurs, and we do that for all of our processes. And the automated DNA sequencer works out all the different lengthed pieces of DNA and puts them into order, the details of which you don't really have to know, but it all works on the different fluorescent dyes being detected by the machine. So the laser beam here is detecting where the different dyes are, and we get a readout, okay, which tells us the sequence in which all the different bases occurred. Okay. Okay, so just in summary, okay, that's quite complicated. Okay, so we've put our primer, DNA polymerase, free nucleotides and modified nucleotides all into a PCR machine. We do lots and lots of cycles of PCR, so we get lots and lots of different size lengths of DNA. Okay, we then separate those lengths of DNA with gel electrophoresis. Okay, and then we can sequence the DNA by sequencing the different lengths of DNA we've got. Okay, so that's how chain termination is done. But the problem with this is it can only sequence up to about 750 bases at a time, and obviously the genome is many millions of bases. Okay, so the rest of this slide, or next of the screencast, will look at how we break down the genome into sizes which can be sequenced. The next slide looks at how the genome is sequenced. Okay, so we've looked at how we can sequence a piece of DNA up to 750 base pairs long. The problem with the genome is it's many millions of base pairs long. Okay, so what we have to do is break it down into smaller bits. Okay, and this is achieved by breaking those bits down by restriction enzymes. Okay, so we break our bits down restriction enzymes. We separate those bits by gel electrophoresis. We then insert each fragment that we've created into an artificial plasmid, which is called a BAC, okay, which I think stands for bacterial artificial chromosome. Okay, it's an artificial plasmid. Okay, so we make recombinant plasmids which have got fragments, individual fragments of different sizes put into each different BAC. We then insert those BACs into bacteria. Okay, transform the bacteria with our engineered BAC in it. Okay, and then we allow the bacteria to multiply, okay, to form colonies of bacteria each, which will have a copy of our BAC with our particular fragment of DNA in it. Okay, and this creates what we call a clone library. Okay, so lots and lots of different colonies of bacteria, each colony with its particular fragment of DNA in it. Okay, then what happened is that laboratories around the world became responsible for trying to sequence particular fragments. Okay, so this was a massive project involving many laboratories throughout the world, and many scientists in each country trying to isolate and sequence small parts of the genome. Okay, and then having broken it down into smaller parts, we then built the picture back up into the whole and how the whole genome looked. Okay, so in terms of once you've got a particular BAC that you're trying to sequence, okay, you isolate that BAC, and then you would remove the fragment of the human genome that you're looking at. You then cut that up again with restriction enzymes into even smaller pieces of DNA, okay, bits of DNA small enough to sequence using our Sanger sequencing, which we talked about in the last slide. Okay, but also what we do is we would treat our back with a series of different restriction enzymes, so we get different fragment patterns. Okay, and we'll see why that's important in a minute. Okay, all our fragments will be separated, okay, by electrophoresis. Okay, and then the painstaking process of using the Sanger sequencing process that we've talked about before to sequence each of these fragments. Okay, the problem with that is that you don't know the order in which the fragments come. You might have sequenced each fragment, but you wouldn't know which order the fragments come in. Okay, so the reason why we don't get different fragment patterns by using different restriction enzymes is that the overlapping regions, okay, which would be illuminated, okay, those overlapping regions can be used to show us how the fragments are all pieced together. Okay, so trying to represent that diagrammatically. Okay, so here we have our large piece of DNA here. Okay, it's all been broken up into fragments. Okay, and these fragments are all here. Okay, and there will be overlapping regions. Okay, so if we look here, here's one fragment here represented by which has been sequenced, and that's our sequence. And here's another fragment here, and there's an overlapping region here, which tells us that sequence runs into that sequence. Okay, and this sequence runs into that sequence there. Okay, so that tells us that this is followed by this, which is followed by this. Okay, another analogy, which hopefully this little thing will work. If I look at the word overlapping here, okay, if I've got the word overlapping here, if I cut the word overlapping after the A, okay, I'll get that word here, these series of letters there, and I'd also have another series of letters there. Okay, the problem with that is if I was then trying to put that together, okay, I wouldn't know whether they went that way round or that way round. Okay. However, if I also cut up the word overlapping, but this time cut it after the E, so this is the analogous to treating my fragment with a different restriction enzyme, what I would get is I would have a different pattern, okay, but again, I wouldn't know which way around these came, okay, so I can't seem to move this, okay, but what we can do, okay, is I can use overlapping regions, okay, to work it out. So I could work out that the RLA there is an overlapping region, okay, and then what I can do is therefore work out that this bit here is that bit there, okay, and this bit here is that bit there, and I can reconstruct my word overlapping. Okay, so that's the principle by which these overlapping sequences are used to work out the order in which the sequences that have been sequenced, the fragments have been sequenced, and the order in which they occur in the piece of DNA you're trying to sequence. Okay, so trying to represent all of that again in an animation, okay, which I hope you'll find useful. Okay, moving on to the last slide. So that's the slide we just looked at how we break the genome down into manageable fragment sizes. We sequence the fragments, and then we reassemble the fragments, the known sequences on the fragments, back into sequencing the whole genome. And again, this took many years with many scientists working in many laboratories throughout the world. Okay, and the next slide looks at, so what? What was the reason for working out the genome? Why is this useful to biologists and medicine and so on? And what I've done is I've listed a few ideas here as to how this could be helpful. Okay, so I'll just very quickly go through these. Okay, so for biology, forgetting medicine for a second, what it's done is it's enabled us to work out how human migration across the world has worked. Okay, so different populations moved throughout different parts of the world throughout history, and by comparing genomes of individuals, we can actually track how different populations have migrated across the globe, because the genomes of closely related people will be more similar to genomes of more distantly related people. Okay, looking at disease. Okay, we can identify genes which predispose people to particular types of disease. So we're finding more and more about the genes that predispose people to heart disease and various cancers and so on by knowing about the human genome. Okay, and obviously if we were able to identify a particular set of genes which contributed to heart disease, obviously we could treat that individual with particular drugs or advised lifestyle approaches to hopefully stave off the onset of heart disease. Okay, similarly with cancers, if we knew you were predisposed to a particular type of cancer, we could scan you more often and be able to pick up the cancer if it ever came about more quickly, and provide the appropriate treatment. Okay, and certainly with treatments of diseases, it's clear that certain drugs, okay, are more efficacious with people that have particular genetic makeups than others, and therefore we could target the right drugs for the right patients to be more effective use of NHS resources and so on. Okay, so those are, if you like, human genomes of why that's quite interesting. Looking at genomes of other species, okay, how this has helped biologists, going back to the stuff you did in F212, looking at evolution. Okay, the comparing genomes of different species lets us see how close species are related to each other. Okay, i.e. the more similar their DNA are, the more similar they're related. Okay, and this enables us to create those diagrams which show the sort of evolutionary tree of life. Okay, and it can be done by looking at DNA rather than just looking at the structures of bodies and so on. Okay. Also, we've also looked at this a little bit. It's helped to illuminate other aspects of development biology, homeotic genes and the homeobox sequences which are present in homeotic genes which bind to DNA. So it's enabled evolutionary development biologists, Evo-Devos as they're known as, with the work that they do and trying to work out how bodies form. Okay, and also there's another example here. Certain diseases which are present in other species, okay, because of the genetic makeup they have, i.e. similar genes, okay, which provide similar diseases in another species, they can provide excellent models, okay, for research. So let's say drug research, if we could find a genetic disorder, okay, which provided a particular disease in another species of mammal, okay, we could use that mammal as a model to do drug research and so on, to see the effect of drugs on the specific disease in that particular mammal, which would obviously have relevance to future treatment of human beings. Anyway, so that finishes our look at the human genome. Okay.